Good morning, grade 12 students, and here we continue with our chapter in immunology after we finished the introduction for the immune system. Here we continue to the immune system to talk about chapter 6, role and components of the immune system on your book on page 112. But before we start, we have to talk about the blood. Blood, as we said, is composed of the plasma, which is the serum, and is composed of cells. Plasma, this one composed about 55% of the blood. It contains water, salts, sugars, and it contains mainly antibodies. So whenever we talk about serum, we are talking about antibodies. And the cells that comprise about 45%, they are composed of many components. The first component is called the erythrocytes, the red blood cells. Erythrocytes, the red blood cells, they carry the oxygen and carbon dioxide. So the red blood cells, they have an important role. They carry oxygen and carbon dioxide. We have the platelets that we said they have a role in blood clotting. And number C, we have the leukocytes, the white blood cells. They have a role in immunity. Leukocytes, they are divided into two categories, something called myeloid and lymphoid. So let's repeat this. We said that the blood is divided into two important parts, the plasma and the serum and the cells. So the plasma, it's about 55%. 55% it contains water, salts, sugars, and antibodies, as well as the cells, they are composed of 45% of the blood. They are composed of erythrocytes, the red blood cells. They have a role in carrying oxygen and carbon dioxide. The platelets, they have a role in blood clotting. And we have the leukocytes, the white blood cells, they have a role in immunity. Leukocytes, they are divided into two parts, myeloid and lymphoid. Myeloid, they are divided into three main components. Myeloid, we have the granulocytes. As we said before, they have multi-lobed nucleus. Under the category of the granulocytes, we have three different kinds. We have neutrophils, eosinophils, and basophils. Granulocytes, I repeat, they are composed of three main types. As we said, they have multi-lobed nucleus and they have granules in their cytoplasm, as we said in the introduction. So granulocytes, they are divided into three different kinds. Neutrophils, they contain neutral granules. So neutrophils, neutral. So here is the first one. Neutrophils, neutral granules. Eosinophils, so they have acidic granules. Basophils, they have basic granules. The second type of the myeloid is the monocytes. As we said before, they have horseshoe-shaped nucleus and they become mature macrophage and they engulf the microbes or the pathogens by the process of phagocytosis. Mast cells. This is a new one, the mast cells. Mast cells, they have round nucleus and they secrete histamine. This have a role in allergy. So the mast cells, they work in the case of allergy by secreting their histamine. So this is the mast cell. So I repeat, myeloid, they are composed of three main components. The first one is called granulocytes. As we said before, they are cells that having the multi-lobed nucleus and they have granules in their cytoplasm. Neutrophils, they contain neutral granules. Eosinophils, they contain acidic granules, and basophils, they contain basic. So neutrophils, neutral, basophils, basic. Eosinophils, هي الوحيدة different, so it's called acidic granules. About the monocytes, they contain the horseshoe-shaped nucleus, and whenever they become mature, they can engulf the microbes by the process of phagocytosis. Number three, it's called the mast cell. The mast cells, they have round nucleus and they secrete histamine and they play an important role in allergy. So this is about the myeloid. Moving now to the lymphoid. Lymphoid, we have two important main components that we talked about them before. We talk about the B lymphocyte and the T lymphocyte. B lymphocytes, whenever they mature, they become something called plasmocyte. What the meaning of plasmocyte is the B lymphocyte when it's mature and it's able to secrete antibodies. So whenever the B lymphocyte they mature, they become something called plasmocytes. Why they be called plasmocytes? Because they secrete antibodies to neutralize the antigens. What the meaning of neutralize the antigens? To destroy or to kill the non-self antigens. However, the T lymphocytes, we said that the lymphocytes they are called killer cells. So they are composed of two types of cells. They compose of T helper, T8 killer. So T4 helper and T8 killer cells. What is the difference between these two? T4 helper, it secretes cytokines. And remember this word, we're going to talk about this in details in the coming sessions. Cytokines. However, T8 cells, they are called killer cells. They secrete two substances, perforin and the granzymes. What's the meaning of perforin? Remember when we talk about the T cells, they perforate, they cause the perforation of the cells, they cause pores in the cells. These pores they will lead to the destruction of the cell, of the infected cell. So we cause it to the pores, it causes perforation. This perforation will cause to be holes inside the cell. These holes inside the cell will release the component of the cell. Whenever the components are released, this will lead to the death of the cell. Then the granzyme becomes, and it will kill, it degrades the cell or it degrades the infected cell. So I repeat, B lymphocytes, we said that they become mature plasmocyte and they secrete antibodies. These antibodies, they have a role in neutralizing the antigens and killing the non-self antigens. And T lymphocytes, they are composed of both T4 helper and T8 killer. T4 helper, they have an important role in secretion of cytokines. We're going to talk about cytokines in details in the coming sessions. And T8 killer, T8 killer, they kill the other cells by using two important weapons. These weapons, they are perforin and the granzymes. Perforin, as its name, it creates a pores inside the cell. This pores will cause the release of the component of the cell and its death. Granzyme comes and degrade the cell and kill the infected cell. So here you have to know that antibodies, they are secreted not by B lymphocyte, by mature B lymphocyte. They are called plasmocytes. So now let's move to document 1, HLA, a major self marker, on page 114 on your book. So HLA is self marker, major self marker. So a self is determined by set of molecules expressed on the membrane of the cells, and every one of these is specific for each individual. These represent the biological identity marker, which is unique for each individual, and they are called self markers. What are these markers and what is their importance? Let's start with document A. We have evolution of different types of skin graft in the mouse. First one, we have the autograft. Autograft that is done within the same body. 2 days later, the aspect of the graft showed vascularization appears around the graft. It's become pinkish in color. Vascularization, it means the blood it can pass through the region where the graft occurs. 1 week later, the graft is integrated into the neighboring cell. So it moves into the neighboring cell, and 15 days later, the graft is accepted. Why? Autograft, it occurs within the same body. So within the same body, the cells they have the same HLA. While isograft, the graft that's done between two identical twins, the graft is accepted also. So 2 days, vascularization appears around the graft, it's integrated, and finally the graft, after 15 days, it's accepted. While allograft, a graft that's done between two different individuals that belong to the same species, both of them they are mice, but one of them is white and the other one is gray. So vascularization appears after about 2 to 3 days. 1 week, redness and the uh, edema appears around the graft, and then the graft it turns black, dry, and finally the graft is rejected. So the graft here it's rejected after about 15 days. This since we have different HLA. Document B show the percentage of transplant survival in two different studies. So here, compatibility of kidney transplant between the donor and recipient. So here we have HLA2 classes, HLA class A and HLA class B, and HLA class DR. Okay, so here it's the organ transplantation in a medical is a medical intervention whose goal is to replace a missing or a deficient organ by a healthy one. Two statistical studies were carried out in order to determine the rate of survival relative to the number of incompatibility. So whenever they have incompatibility in the HLA, as the incompatibility increases, the graft acceptance will decrease in percentage. So the first study, which is the Eurotransplant, determined the number of incompatibilities. Incompatibility is the opposite of compatibility. So between the donor and the recipient at the level of HLA class A and HLA class B loci. So we have two classes now of HLA, HLA class A and HLA class B. The second study, it's showing Oxford. It determines those at the level of the HLA class DR loci. So HLA class A, HLA class B, this is the blue one in the European transplant, and the Oxford transplant, HLA class DR. So here, incompatibility number, it's showing here that the grafting, it depends on some type of HLA and not all the types of the HLA. Organization and the expression of MHC. Major, whenever we talk about MHC, you are talking about major histocompatibility complex. So these MHC genes, they code for two classes of membrane glycoproteins. Glycoproteins, these they differ by their structure, their distribution, and their role. These membrane molecules have been identified on the membrane of leukocytes, and they are called human leukocyte antigens, HLA. These HLA molecules of classes 1, which are coded by loci, they have different positions. They have A, B, and C. They are expressed, as we said before, on the nucleated cells. Class 2 molecules, which are coded by the loci DP, DQ, DR, they are expressed by some cells of the immune system. So till now we have HLA class 1, we have 3 different loci or 3 different types if you want, class A, class B, and class C, and HLA class 2 coded by DP, DQ, DR. They are expressed by some of the new cells. These genes coding for HLA molecules, they are highly polymorphic. What the meaning of highly polymorphic? They are highly different and they change from one individual to another. The genes coding for HLA molecules, they are highly polymorphic. That is, each exists in large number of allelic forms. However, each individual has 2 alleles of each gene, and every individual has 2 alleles of every gene. One of them is paternal, and the other one is maternal. So one of the fathers or the male's origin from the father, and the other one maternal is from the mother's origin. The expression of the MHC, and pay attention to this word, expression of MHC is co-dominant. So both of them will be expressed. Is the case of co-dominance, where both alleles will be expressed. So each individual expresses on his cell membrane the proteins corresponding to both alleles of each gene. This is what's known by the expression of MHC. So what I want from this probing the documents, referring to document A, throw out the definitions of the following terms: autograft, isograft, and allograft. Part 2, what can you deduce from documents A and B? Let's start step by step. Already we know that autograft is the graft that is done within the same individual. Isograft done between two identical twins. Allo is the graft that's done between two genetically different individuals belonging to the same species. Like, for example, the third experiment here, in the case of the allograft, two different genetically different individuals, but they belong to the same species, which is the mouse. And I added the fourth one, which is the xenograft. Xenograft, graft between two genetically different individuals belonging to different species. Here, I aimed to do interpret. I interpret the experiments of documents A and B. They want here, what can you deduce? They only want to deduce. So I added for this the interpret. So after applying an autograft or an isograph between the mice having the identical HLA or identical MHC, by 2 days, vascularization appears around the graft. 1 week later, it's integrated into the surrounding region or the neighboring zone, and 15 days later, the graft is accepted. However, at the same time, after applying an allograft between two mice that have different HLA or different MHC. So 2 days later, vascularization appears around the graft. 1 week, redness and edema. So redness and edema swelling will appear around the graft. 15 days later, what happened? The graft will be rejected. It turns out to be black and dry. What does this indicate? This indicates that the acceptance and the rejection of the graft, it depends on one factor. This factor is the similarity of the MHC molecules between the donor and the recipient. However, in document B, when the incompatibility number of HLA class A and B between donor and recipient is zero, what do you say what of incompatibility? Incompatibility meaning that they incompatible, they are different. So percentage of incompatibility was zero. So they are 100% compatible, meaning that they are 100% similar. Pay attention to this. Incompatibility meaning that they are different. Compatible, they are similar. So when the percentage of incompatibility is zero, what does this mean? This means that incompatibility is zero, so 0% difference. So 100% they are compatible. So the percentage of the graft survival after 1 year is about 72%. As the incompatibility number of HLA increases to 4%, the percentage of graft decreases slightly to 60%. At the same time, when the incompatibility number of HLA class DR between the donor and recipient is zero, so 100% compatible, the survival percentage is 85. As the incompatibility increases to 2, the percentage of the graft it decreases and drops to 55%. What can we deduce here, or before we deduce, that is the indication? This is the graph depends on the similarity of HLA class DR more than class A and B. Why? Because it drops to 55% when the incompatibility increases to 4. So it drops to the half. So acceptance and rejection of the graph, it depends on some type of the MHC between the donor and recipient. So acceptance and rejection of a graph, it depends on the degree of similarity of MHC molecules between the donor and recipients. And to be more specific, it depends on some kinds of MHC, like the case of HLA class DR. Why? The class DR, when it increases the incompatibility to 2, the percentage of the graph, it decreases to 55. So it decreases into half approximately when the incompatibility becomes 2%. So they are compatible 98. So 98 compatibility, even though it decreases to about 55%. Here we finish everything related to the self. Now I'm going to move to the non-self, which is document 3. Document 2 for this year is cancelled. So document 3, the non-self, page 119. Document 3 talking about the non-self. Non-self is everything that does not belong to my body, and they have different HLA. So everything that doesn't belong to my body is classified as the non-self. So here, infections against an organism, pathogenic agents. Pathogenic, they can cause a disease. So immune system is able to distinguish between the components to one's body and others that are foreigners that doesn't belong to our body. What are the different types of the non-self? What are the different types of the foreigners? Let's start first with the pathogenic agents. Infection agents are organisms that can cause infection, that can infect other living organisms, that is, invade other organisms and develop at their expense. They can cause disease, and whenever they can cause disease, these agents are called pathogens. In Latin, pathogen, yani disease, generator, so generator of a disease, that's why it's called pathogen. It can cause a disease. They include some viruses, bacteria, fungi, parasitic protozoa, and some worms. All except worms. They are microscopic, meaning that they are seen under the microscope, called microorganism, or simply they are called microbes. Some infectious agents they are transmitted from infected person to a healthy one, either by skin contact, by the contact of the skin, by the sexual contact through the sexual intercourse, through the blood, or from a pregnant woman to their fetus. Other agents they are transmitted indirectly through air, water, or food. So let's talk about some infectious agents and the diseases they cause. Infectious agents, we have some of them, we have the viruses. Viruses, we have a lot of viruses. For example, we have the influenza virus, the measles virus, the mumps virus, the vertical zoster virus, the human immunodeficiency virus, HIV. Influenza, it can cause the flu virus. The measles, it causes the measles. Mumps, it causes the mumps. The vertical zoster virus, it causes the chicken pox. The human immunodeficiency virus, HIV, human immunodeficiency virus, it causes the disease which is called the AIDS, acquired immunodeficiency syndrome. AIDS, A acquired, I immuno, D deficiency as syndrome. Bacteria, we have a lot of bacteria. We have streptococci, salmonella, we have vibrio cholera, we have Clostridium tetani, and we have the Mycobacterium tuberculosis. So, streptococcus can cause pneumonia. It can cause, for example, salmonella. It causes typhoid fever. بيسموها التيفوئيد. Vibrio cholera, it can cause the cholera. Clostridium tetanus, it can cause tetany. It can cause the tetanus, and tuberculosis, it can cause the tuberculosis disease. You want to ask me, do you have to study these or not? You don't have to study this. Fungi, protozoa, and worms, they can cause mostly malaria, for example, malaria, they can cause intestinal infection and mouth lesions. This is the influenza virus in document B that is shown under the microscope. The elements of the non-self. Cells of the immune system, they are able to recognize all the sorts of the non-self molecules that enter the body. These are molecules, they are called the antigens. Antigen is a large molecule. Usually it's called either a protein or a complex carbohydrate. It could be a component of a cell, such as a bacteria, a red blood cell, or a nucleated cell, or it can be a particle, such as a virus or a pollen grain. Everything that enters the body and is different from our body, either it's a protein, it's an antigen for sure, either a protein or it's a part or a component of a carbohydrate, which is a complex carbohydrate. Here, we said that it can be a part of a cell, or it can be a particle like a virus or polymerase grain. An antigen could also be soluble, that is in the form of a free soluble, such as toxin released by bacteria or presence in the venom of animals like snakes and bees, or it can be a harmless substance experimentally injected into the body. So here you have to know that the immune system is not only directed against toxin and pathogenic, but also against any non-self. So any non-self that enters into my body, the immune system will be able to recognize it and to attack it. Here, whether harmful or not, it will attack it, including vaccines, some of the vaccines, or even though all of the vaccines, tissue transplant, red blood cells from an incompatible blood group. Incompatible blood group is the group that does not fit with my blood group. The self might undergo certain modifications. Our cells, some of them, they undergo some modifications. These modifications are called the mutations, and they will cause to the appearance of something called the modified self. Example about this, the cancer cells. Cancer cells, they are recognized or they are identified as modified cells. They are cells inside their body, but they undergo a mutation. These mutations occurs frequently, and this will lead to the appearance of these cancerous cells. These cancerous cells, they are not non-cells. They are modified cells. They are belonging to my body, but they undergo modification. They undergo some mutation, and this will lead to the appearance of these non-self that are called the modified self. Okay, so here you have to know that the antigen is divided into two categories. Either it's cellular, it's found in this membrane of a cell or a virus, and the antigen can be soluble, it's free, it's secreted by another cell, and it's not bound to the cell itself. And you have to know that the antigen can be into three categories also: non-self antigen, modified self, or a self. Non-self is outside the body, it's foreigner. So example about this, the bacterial toxin. A modified self synthesized by our body, but in exceptional condition under the, for example, the case of mutations. An example about this, cancer cell or viral infection. Self, it's sensitized by our cell in the normal conditions. So here you have to know from the documents. I want only part one, referring to the information provided in paragraph 2. Can we consider HLA molecules as antigen? Justify yourself. Yes, we can consider them as antigen because they are proteins that are recognized by the immune system. So they are called antigens because they are proteins and they are recognized by my immune system. For this, we finish document 1 and document 3. Next session, we'll continue with document 4 to talk about the cells of the immune system.